Okay, this is our third lecture on the nervous systems, specifically talking about nervous tissue. And we left off starting to talk about neurons, and in fact, we started to discuss myelinated versus unmyelinated neurons, and that we have cells that produce this myelin, and the job of myelin is to increase the speed in which nerve impulses can travel down the axons of neurons. So we're going to continue on this discussion of myelination. So here we have this myelinated neuron. So here's the myelin, the layers of lipid and protein that insulate that axon. Here's the Schwann cell, so this tells us that this neuron must be in the peripheral nervous system, because in the central nervous system we have oligodendrocytes producing the myelin. Over here are the unmyelinated axons. So again, we still see the Schwann cell, but we do not see any myelin sheath surrounding those axons. So myelination is the production of myelin sheaths. This occurs during fetal development, child development, and it's going to allow an adult to be more coordinated than children. If we were to assess the nervous system in an infant or a child, it wouldn't be functioning as fast as an adult, and this is largely because the myelin is still developing. So myelin insulates the axon, increases the speed of nerve impulses. It produces a white appearance in the tissue. So we hear often that the nervous system has what's called white matter versus gray matter. Well, white matter is largely myelinated axons, or myelinated fibers. So when you get a lot of these myelinated neurons all packed together, the tissue appears white. And it's white because of the lipid. The lipid appears white. If there's low amounts of myelin, so a lot of unmyelinated neurons, the tissue appears gray. So we have what's called gray matter versus white matter. Gray matter is going to be unmyelinated neurons. They send their action potentials a little slower. The neurons are a little smaller, but there's a reason that these neurons don't have to produce fast action potentials. Again, they can be surrounded by Schwann cells, but the Schwann cells are not producing the myelin, at least in the PNS. Now, in the CNS, we can see here, panel A is a transverse section through the spinal cord. If we were cut right through the spinal cord, we could see that there is gray matter, which forms kind of an H shape on the inside of the spinal cord. So this is all gray matter, unmyelinated axons, whereas surrounding the gray matter is white matter. Indeed, the white matter is going to be transmitting the information up and down the spinal cord. So our spinal cord is used to deliver information up to the brain or down from the brain. And that has to happen very quickly. So that's largely the white matter. Gray matter is more analyzing, interpreting where does the information have to go. And that doesn't require the myelination. In the brain, so in panel B, we see a frontal section through a brain. And we see the brain also has gray matter and white matter. The white matter, in fact, is more on the inside, whereas the gray matter is largely on the outside or superficial layer of the brain called the cerebral cortex. There's some gray matter buried inside, but largely you would find most of the gray matter around the outside of the brain. So let's start our discussion on neurons. So again, neurons are the functional unit of our nervous system. They have the capacity to produce action potentials, which again, we're going to be discussing action potentials in class, but an action potential is a nerve impulse, electrical current traveling down the axon. So this is a nice basic drawing of a neuron. It's called a multipolar neuron. We'll talk more about what that means, but multipolar means there's many processes sticking off of the cell body. These processes here are known as dendrites. So these are smaller little projections off of the cell body, which I've circled. Cell body contains the nucleus. So this neuron is going to be receiving stimuli. And part of the neuron that receives the stimulus could be a neurotransmitter, could be pain. We could have pain receptors on this neuron. But the dendrites receive stimuli. Cell body houses the nucleus and other organelles, and then the axon is the longer process that conducts the action potentials, or transmits the action potential down this long axon. And then we're going to learn that once this action potential travels down the axon, there are end bulbs, which should sound familiar, that release neurotransmitters. Neurotransmitter would be coming out of all these little tiny end bulbs. So this could be a somatic motor neuron, where these neurotransmitters are acetylcholine. So those are the three basic parts to a neuron. This drawing includes some of these other parts. So let's see what else there is. So dendrites, cell body, axon. We can find all those on this drawing. Dendrites, cell body, and then the long axon. Missile bodies are small little pieces of the ER, which we're not going to talk too much about. Neurofibrils are little intracellular fibers we're not going to really talk about. The axon hillock is a cone-shaped area between the cell body and axon. So this is basically where the axon is going to start to come out of the cell body. And then on part of that axon hillock is what's called the trigger area. The trigger area is where an action potential is going to first form. So we have the axon hillock right here, and somewhere on that axon hillock is where we're going to form an action potential. And that action potential is going to travel, and the arrows represent the direction in which an action potential is going to travel down the axon. Notice we can also see here that there are Schwann cells surrounding the axon. So these are the little nuclei of the Schwann cells. Underneath the Schwann cell is going to be the myelin sheaths. So we see the myelin sheaths between the Schwann cell and the axon. But notice there are some gaps. Gaps in between the Schwann cells. These gaps are called nodes of Ranvier. And we'll talk more about nodes of Ranvier in class. And then eventually at the terminal end, the axon terminal, we have end bulbs. And we know what's in the end bulbs. Remember what's in an end bulb. Synaptic vesicles containing neurotransmitter. Now, are all neurons containing acetylcholine? No. That's just one type of neurotransmitter for a particular type of neuron. So these are parts of a neuron. We're going to be discussing a lot of this stuff in class. Next thing we can do is classify neurons. So there's different types of neurons. And this is going to classify our neurons based on structure. Structure of the neuron, not necessarily functional classifications, which we'll do next. So there are multipolar neurons. Multipolar means many processes, many things sticking off of the cell body. So there could be several dendrites and maybe one axon. So panel A represents a multipolar neuron. Here's the cell body, and then here are the many dendrites sticking off of the cell body, and then one single axon. So most motor neurons are multipolar, kind of like our somatic motor neurons that control skeletal muscle. And most neurons in the CNS are multipolar. So collectively, most of our neurons that we find in our nervous system are multipolar. There are others that are bipolar. So bi means two. So two processes that come off the cell body. So in panel B, we have bipolar neuron. Notice here's the cell body. And there's only two processes. There's one dendrite and one axon. Now we don't care about all these up here and these down here. We're looking at just how many things come off of the cell body. If there's two, it's called a bipolar. A lot of our neurons in our special senses are bipolar. So special senses, which we're going to learn about later in the semester, include vision. So our retina of the eye contains bipolar. Olfaction, which is smell, are bipolar. And then many neurons that allow us to hear are bipolar. Finally, the third type of neuron could be a unipolar. So uni means one. So this is an example of a unipolar neuron where there's just one thing sticking off the cell body, but it often then splits into many processes. So again, we're just looking at the cell body, how many things come off of it. If there's just one, it's a unipolar. These are most of our other sensory neurons. So sensory neurons in the skin, sensory neurons in our muscles and organs in the body are often unipolar. So most of those skin sensors are unipolar neurons. Okay, next thing I want to do is talk about functional categories, which we've already done a little bit. So remember, you can have sensory neurons. Sensory is a function. So they often have receptors on their dendrites, and they detect the stimulus in the environment, whether it's our internal environment or external environment. Once these sensory neurons detect the stimulus, they change that stimulus into an action potential, and it goes to the CNS. So remember, if it's going to the CNS, we also call these afferent neurons. These neurons will then go into the CNS. They will either synapse. If you remember what synapse means, it's a connection, or when two things come very close to one another. These sensory neurons either synapse with a motor neuron or an interneuron. Most sensory neurons are unipolar. Again, mentioned that on the previous slide. That's an anatomical or structural category, unless they're in the special senses. Eyes, nose, ear. Those would be bipolar. So notice for every neuron in the body, you can categorize it two ways. One, on its structure, multipolar, bipolar, unipolar, and by its function, sensory, interneuron, or motor neuron. So interneurons, also known as association neurons, are multipolar neurons of the CNS, so only found in the CNS, and these interneurons are analyzing, they're integrating, they're figuring out where the information needs to go. So they're going to send signals to the brain, or they're going to say, alright, well we need to tell this motor neuron over here, so they're going to synapse with a motor neuron. 90% of our neurons in the body are interneurons, because most of our brain and spinal cord is made up by interneurons. Third functional category are the motors. The motor neurons are going to send the information from the central nervous system to an effector. So motor neurons are controlling your muscles, your glands. These are all structurally multipolar neurons, and again they're also known as efferent because we're leaving the CNS and going to an effector. So these are the three functional classifications. This just shows some examples. So if I were to ask you, and let's say we covered this up, and I asked you what kind of neurons these are, structurally we would say unipolar, 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 and unipolar. Functionally, they are sensory neurons. Oops, sorry about that. So these are types of sensory neurons. They're all found in the skin. So panel A is a Meissner corpuscle. They help with our sense of touch. Panel B has these Merkel discs. These are also sensory neurons in your skin. Here's a Pacinian corpuscle, which you remember we talked about that in the skin. And then panel B is a nociceptor, which is pain. So these neurons sense pain. So that's the end of our third video. So when we come into class, we're going to be picking up on slide 17.